were cyanobacteria. So as we mentioned, it's sort of unexpected that cyanobacteria are major nitrogen fixers. And why was that? Who remembers from last time?
They're very associated with oxygen, and nitrogenase, the enzyme, can't really function in oxygen.
Exactly. In fact, it's destroyed by oxygen. So some cyanobacteria have evolved specialized cells to get around this problem. This is actually a picture of one of the organisms I studied during my dissertation research, and you can see that it has this big long filament, and that at the very end there's this weird looking cell. This is the same kind of cell that you see in this picture here by another species called Anabaena. These are the photosynthetic cells, and they look very kind of round, normal. These elongated cells, these are called intercalary heterocysts, and this is called a terminal heterocyst. So in many of these filamentous cyanobacteria, you end up with a specialized cell. And what this allows you to do is have a different oxygen environment in here versus over here where the oxygen is being pumped out. And that's sort of illustrated here on this diagram. So essentially, these cells are fixing carbon, they're producing oxygen. In this cell, there's no photosynthesis going on, or at least none of the oxygen production portion of it. And so essentially what's going on here is the nitrogen fixation, converting N2 to ammonia. This requires a lot of energy, as we're going to come to in a second, though, right? So you need a lot of ATP to fix nitrogen. And in order to accomplish that, you need carbon to make this work, okay? So essentially the fixed carbon that gets used over here and the ATP that gets used over here is translocated into the heterocyst, where it can cause, it can feed the nitrogen fixation. Okay. And then the nitrogen needs to be distributed to the so-called vegetative cells here, okay? And so that's usually in the form of glutamine, okay? So the NH3 gets converted to glutamine, moved over, and then glutamine is readily incorporated into other amino acids or many different kinds of, yes. So, but it turns out not all cyanobacteria that fix nitrogen are filamentous. Or not all of the filamentous ones produce heterocysts. So for example, trichodesmium, which was one of the earliest studied examples. This is a common marine cyanobacterium that looks like this under the microscope. They don't fix nitrogen. They do fix nitrogen, but they don't have heterocysts, which is sort of question, right? So what do? Do they exclude oxygen in the middle of this, right? Do they exclude oxygen in the middle of these bundles? Seems unlikely because oxygen should diffuse right through. But then about, maybe 20 years ago now, a researcher up at UC Santa Cruz, John Zare, discovered that he started looking for NIF-H gene expression in the ocean. Now, NIF-H is one of those key enzymes that's involved in the production of nitrogenase, this gene. So the nitrogenase gene expressed all over the ocean. And when he looked for the specific cells, it wasn't trichodesmium, it wasn't heterocystis guys, it was these little unicells. And so the question is, how is this possible? How are they protecting their nitrogenase? And we honestly don't know. So this is one of a very active area of research that his lab and others are still pursuing. That said, if you want to fix nitrogen in an anaerobic environment, no problem, right? So there's many anaerobes, for example, things like Clostridia and others, sulfate reducers that are nitrogen fixers in the sediments. And they don't have to worry about protecting their nitrogenase because it's no problem. Okay. All right. So nitrogen fixation, though, of course, is crucial. And we've talked about the symbionts that do it. We've talked about the fact that many phototrophs like cyanobacteria do it, but also many other heterotrophic groups do. But one of the things we need to think about in terms of nitrogen is the whole cycle together, right? And of course, the loss of all of that nitrogen that has been fixed or laid down as fertilizer is a real problem if you're a farmer. So we've talked about algal blooms, right? We've talked about runoff from farms and how that contributes to the problem of eutrophication, right? But another problem is if you have floods, especially in the springtime or thunderstorms in the early summer, if a lot of rain falls in a short amount of time, it'll flood fields. When that happens, it creates anaerobic conditions, which really favors denitrification. And since the fertilizer placed onto farms by farmers is usually in the form of nitrate, then we end up with our nitrogenous intermediates, which, as we were reminded last time, can be greenhouse gases, as well as N2. Okay, so this would be a negative here. But as we're going to talk about in our next lecture, which will begin today, it's a positive if you want to get rid of nitrogen from systems like in wastewater, right? So there's a lot of carbon and nitrogen in pee and poop, and so we got to get rid of it. So that's actually something we'll talk about as well. Now the opposite, remember, ammonia oxidation, or as it's referred to here, nitrification, is the conversion of reduced ammonia like NH4+, or you can, when you see this, you can think NH3 as well. I think somebody stayed after class and asked about that last time. If you see NH4+ or you see NH3, they're essentially interchangeable in my class. Not in chemistry class, but in this class they are. Here we're oxidizing these through a series of intermediates. This is also bad for farmers though, because not only can the nitrate be denitrified, okay, but also nitrate is more soluble than ammonia. So it is more likely to run off into, creeks and streams, rivers. And the other thing is if you do have a charged form, the ammonium ion, then this can actually bond. It can form an ionic bond with negatively charged clay particles in the soil. So in this case, it does matter that it's charged, okay? All right. So those are just some kind of applied things to think about. All right, so now let's move on and talk about sulfur, the other major cycle we'll have time to talk about this semester. Like nitrogen, you have highly reduced. This is rotten eggs. This is hydrogen sulfide. And then we have highly oxidized sulfate, right? And we talked about both of these, right? So you can oxidize reduced sulfide and you can reduce sulfate the other direction, okay? And we're going to talk about those. So it's pretty abundant in many environments, especially the ocean, right? So it's the second most abundant ion in the ocean, anion in the ocean after chloride. So we all rely on it because it needs to be assimilated because we have essentially sulfide containing amino acids like cysteine and methionine, right? But hydrogen sulfide is this gas which is actually toxic to many organisms. Unfortunately, it has a real tendency to bond. It binds very tightly with metals. So as we'll talk about with our bioremediation, that actually is kind of good because it sequesters. So sulfide can trap metals and make them precipitate. But when it comes to cells, We have things that have metals like iron in them inside our cells, and they have them inside bacteria and archaeal cells as well. So things like cytochromes, if they encounter sulfide, can be bound up to that and they're going to be inactivated. So that would be bad. Okay. So organic forms, like I mentioned with cysteine, methionine, are going to be sulfhydryl groups, but you can see this is reduced, and so it's essentially the sulfide form. So let's talk about the reservoirs like we did with carbon and nitrogen. Not quite as large and atmospheric, which is good, because can you imagine if we had lots of sulfur in the atmosphere? In fact, this was the case on the early atmosphere. There was much more sulfide back then. And as we'll talk about, that's not good. Okay. So in the atmosphere, it's sulfur dioxide. And at the end of this portion of the lecture, we'll find out why having a lot of oxidized sulfur in the atmosphere is bad for life. In the ocean, as we said, sulfur, and mostly going to be sulfate, is very common. Okay. Incidentally, hydrogen sulfide is a gas, right? So it's going to go up in the atmosphere, but in the presence of oxygen, it will be oxidized to sulfur dioxide very rapidly. Okay. So huge amounts of sulfur in the oceans, even larger amounts in rocks and sediments, but of course that's going to be unavailable to life, just like we saw before. Because we all have sulfur in us, organic material is a pretty large pool, okay? Both in terrestrial and oceanic biomass. If you pool these together, you end up with large numbers. So what are the key processes? So we've already talked about these in our metabolisms. So we've got sulfur chemolithotrophs. So these are going to oxidize hydrogen sulfide to elemental sulfur and then sulfate. Okay, this is an aerobic process. There's also anaerobic photosynthetic organisms. So these are chemosynthetic, but anaerobic phototrophs, like the single photosystem types we learned about in the metabolism lecture, purple phototrophs, green sulfur phototrophs, things like that. These guys are going to also oxidize hydrogen sulfide. Remember, they use H2S as their electron donor, much like cyanobacteria and plants use water. H2O, H2S, right? Okay. There's also some anaerobic chemolithotrophs that function in a slightly different way, but we're not going to go over that. Sulfate reducers, we've talked about these repeatedly this semester. These are found in both bacteria and archaea, but the majority of them are in the group of delta proteobacteria. And that's groups like these desulfovibrio, or sort of the classic lab rat sulfate reducers. Some organisms, though, do not. Oh, I guess I should be showing you the delta G. So this is very energetic. Okay, that's good. Sulfate reduction, we've talked about, is not as energetic, but it's still a negative delta G. So you can still get some ATP. Remember, negative delta G means ATP. Okay. So this is taking the oxidized form and putting electrons on that. This is taking the intermediate. If you remember, sulfur is the intermediate in this step. You don't get as much energy out of this. I don't think I have the delta G for this one, but it's not quite as much. Okay. But these guys often live with these guys. Then there's this weird thing called disproportionation, where you take an intermediate that's partially reduced, the S2 part, partially oxidized, the O3 part. This is called thiosulfate. So thiosulfate can be split or so-called disproportionated. This can be converted into a reduced sulfur component, hydrogen sulfide, and an oxidized sulfur component, sulfate. And this yields a little bit of energy, slightly negative delta G. Most organisms, microbes that do this, do not do this exclusively. So for example, desulfovibrio is a sulfate reducer, but under certain conditions where sulfate is absent, but thiosulfate is present, they can switch over. This is just another example of the metabolic flexibility I introduced a few lectures ago when we talked about metabolism, okay? And then these other things we don't really need to worry about. All right, so like with the carbon and nitrogen, we have aerobic or oxic processes and anoxic processes. So sulfate reduction is going to be an anaerobic process. Okay, so taking sulfate and converting it to hydrogen sulfide, sulfate reduction is anaerobic. Sulfur reduction is also anaerobic. Disproportionation is also anaerobic. Okay. But interestingly enough, the sulfur disproportion does generate that oxidized form, even though it does it under anaerobic conditions. Now, I mentioned that the chemolithoautotrophic sulfur oxidizers, right? These are things like I showed you a picture of Beggiatoa a few lectures back. These are chemolithotrophic organisms. In the presence of oxygen, they can fix carbon based on the energy that they get from converting sulfide to sulfate. Okay. All right. And then assimilation occurs both aerobically and anaerobically. We all need sulfur. Now, there's a global sulfur cycle, just like there was everything else. And just like we said with CO2 and methane, human impacts matter here. So it turns out that when we burn, particularly if we burn coal, right now this is becoming less common as we move towards green energy. But if you burn coal, it releases not only CO2, but it also releases sulfur dioxide. Okay. You actually, the reason we have catalytic converters on cars is also to reduce the release of sulfur dioxide. So in your catalytic converter, the reason people steal them is they have precious metals like palladium in there. And the metals bind the sulfur. We said metals and sulfur can bind together. And it keeps it out from going up in the atmosphere. The reason we care about this, and I'll touch on this in a few minutes, is that before you guys were born, back when I was in my infancy in the 70s, there was a huge problem of acid rain. And this was largely a result of automobiles and factories. So this was a big deal in like the upper Midwest and northeastern US and that part of North America. And when you have sulfur dioxide or oxidized sulfur in the atmosphere, this can lead to acid rain, which acidifies water, which is particularly bad in freshwater. The buffering capacity of the ocean makes it not a big deal. But in lakes and streams in the Northeast and in parts of Canada, they were really impacted by this. Incidentally, sulfur dioxide is also released by volcanoes. And so on the early Earth, there was a lot of sulfur dioxide in the atmosphere because of the high degree of volcanism. And we do still have problems with this. So, you guys may have, seen in the news, like an Icelandic volcano erupts and it causes like air traffic to be disturbed, but you can actually measure detectable sulfur dioxide and things like that in the atmosphere near there. And if we have a major volcano that goes off, this can actually lead to global catastrophes and global acid rain. And that has happened in the Earth's earlier history and likely contributed to mass extinction events millions of years ago. We're hoping it doesn't happen anytime soon. All right, so sulfur does, it exists in the atmosphere, it exists in the water and on land. Interestingly enough, one of the ways that sulfur goes from water up into the atmosphere occurs in the ocean via sea spray. So there's things like DMSO, dimethyl sulfoxide, which has sulfur, which can be ejected up into the atmosphere when wave action stirs up the oceans. And we have a large amount of release of artificially through both volcanism and humans. It can also then be rained out again. All right, so let's have our first poll question. So which of the following sulfur cycling metabolisms is most likely to occur at the interface between anoxic or anaerobic and oxic conditions? Would it be heterotrophic sulfate reduction, chemolithotrophic sulfur oxidation, heterotrophic sulfur disproportionation, or phototrophic sulfur oxidation? Based on what I showed you a few slides ago, what would be the best answer? So what is it that is going to occur right where you see that interaction of both the anaerobic and the aerobic conditions? All right, is that everybody? I think it might have been a little bit tricky. So a lot of people put A, B, and C. So I'm going to go back to that slide. I think maybe people were a little tricked by this. So if you put sulfate reduction, maybe it was because you saw sulfate here. So sulfate is produced under aerobic conditions or oxic conditions. But typically the sulfate reduction occurs only under anaerobic conditions. And I also said that sulfur disproportionation only occurs here. So, excuse me, the answer I was looking for is this reaction. Sulfide produced anaerobically percolates up, and where it is liberated up into, like, let's say the water column. Like, think about hydrothermal vents where chemosynthesis is very common. Sulfidic waters bubble up. This provides hydrogen sulfide. It mixes with the fresh water, the seawater that is oxidized, that has O2 in it. And it's at that interface where the vents are leaking the sulfide, but then there's oxygen in the water that this can occur. Okay, so the best answer was actually B, chemolithotrophic sulfur oxidation. Okay. And phototrophic sulfur oxidation is an anaerobic process as well. So this is the one that requires oxygen. Okay. All right. So I've already alluded to acid rain, so I'm not going to belabor this. It turns out that acid rain includes both sulfur oxidized forms and nitrogen oxidized forms. And we put an X here because it's a mixture. So it can be SO2, it can be SO3, NO2, NO3. Okay. So all of these contribute to acid rain. And acid rain still goes on, particularly in countries, especially developing countries that have industrialized more recently. We've forced clean energy, sort of clean processes onto factories in North America because we had this problem decades ago. So they actually put scrubbers and things like that on the stacks to try to stop the release of the oxidized, and as I mentioned, catalytic converters on cars. But in some countries, the regulations haven't caught up with that because they're industrializing now as opposed to 40 years ago. So, and essentially what you're making is sulfuric acid and nitric acid, and that's why we call it acid rain. And if it falls onto fresh water that doesn't have the pH buffering capacity of the ocean, then you actually, this led to serious like fish kills and like almost like killing off of all of the animals and vegetation living in some lakes. Okay. All right. Any questions about sulfur or nitrogen cycling?